Hello and welcome to Dawn or Doom. I have the honor to introduce Herman O. Sintim. He is the drug discovery professor of chemistry at Purdue. His research interests include the chemical biology of bacterial virulence, it's a hard word to say, and biofilm formation, isothermal detection of disease biomarkers, and synthesis of drug-like molecules. Born in Nutafo, he grew up in Koforidua, Ghana. He studied at University College London and Oxford and held postdoctoral positions at Oxford and Stanford before joining the faculty at the University of Maryland at College Park, where he was a full professor before moving his lab to Purdue in 2015. Today he will present a talk titled The Nightmarish Multidrug Resistant Bacterial Infection Era is Closer Than You Thought. Please take a moment to silence all electronic devices, but please don't put them away. We hope to see you tweeting to the hashtag Dawn or Doom. Thank you.
Thank you, and also thanks for the audience for taking the time to come today to listen to my talk. And so for today's talk, I hope that I don't scare too many of you. I am just going to present a fact, and then you can make up your mind whether we are at the dawn of something great to happen or we are heading towards doom. And so I'd like to start this talk by giving a shout out to Alexander Fleming, the discoverer of penicillin G. He did a lot to change the world. Before the discovery of penicillin and drugs like penicillin, having surgery could be a matter of death. Soldiers who went to war and were victorious would return home and die from their wound infections. These days, we don't have to think about these kinds of infections that lead to death. And so we have almost become complacent that we don't have any problems with bacterial infections. But as I will show during this talk, it will become very clear that we indeed do have problems regarding bacterial infections. So in addition to Fleming, a few scientists contributed to the antibiotics that we use today. The first person to really make significant contribution to antibiotics is Gerhard Domag. He discovered an antibiotic called Prontosil, which is a synthetic dye. And for that effort, he was given a Nobel Prize for the discovery of the antibacterial effects of Prontosil in 1939. And then following Gerhard were Sir Alexander Fleming, Ernst Chain, and Sir Florey, who discovered penicillin. And we now know that penicillin started a revolution in the development of antibiotics. And for their efforts, they were also given the Nobel Prize in 1945 for the discovery of penicillin and its curative effects. Penicillin, by and large, is not effective nowadays because of drug resistance, but they really set everything moving. And then finally, the last Nobel to be given to the discovery of antibiotics was given to Selma Waksman at Rutgers University for the discovery of streptomycin. And streptomycin was the first antibiotic that was effective against tuberculosis. And so several hundreds of millions of people worldwide carry the TB bacteria. And although TB is not problematic in the US, it is actually problematic in some parts of Africa and Asia. And so you can imagine that with all these great advances and discoveries, it would be very easy for folks at that time to think that we have finally conquered bacterial infections. And so this made the US Surgeon General, William Stewart, to declare in 1969 that the time has come to close the book on infectious diseases. I think it's understandable because we'd already given out several Nobels and several antibiotics had been discovered. And so maybe we could close the book and then move on to other bigger challenges such as cancer or viral diseases, etc. Now, hindsight is always a good thing, but my advice is that irrespective of whatever advance is made, you always have to be careful in making such grand pronouncements. So if we were to look at life expectancy, we can now see that these advances that have been made in medicine and also technology now means that we are living about twice longer than our ancestors. So we can see here that for a very long time, the life expectancy was around the 40s. And I just turned 40 recently, so it meant that maybe a century or 2 centuries ago, I might be thinking about my will really seriously. But I now look forward to another 40 or more years being alive. But we can see that there has been spikes where life expectancy really was lower than expected. And even recently, there was a spike that life expectancy was lower than expected. And the reason is that those periods actually coincided with outbreaks of diseases. And so viral diseases have been very problematic. And so we can see here that during the influenza epidemic of 1918, life expectancy actually went very down. Now, we have heard of several viral outbreaks and other diseases. And so the recent one that comes to mind is the Ebola virus that luckily for us, we were able to contain. But it gives you an idea that even in modern times, we can still have outbreaks, and these outbreaks can actually adversely affect us. Now, these life expectancies are not only, the rise in life expectancies are not only limited to Western worlds. All over the world, even if you look at developing nations, you can see that life expectancy is actually going up. And so it's a great time. It's a great time to be alive, and it's a great time to celebrate. But it is also time for us to reflect and look at history. And so I did talk about the influenza virus outbreak, but there's been times in the past where bacterial outbreak also wiped significant proportion of the population. And so there are examples of the plagues. The infamous one is referred to as the Black Death, and it wiped between 75 to 20 million of people in Eurasia. It turns out that the Black Death was caused, most likely, we can never be definitive for something that happened way back, but from sequencing the genome of remnants of bacteria in skeletons that were excavated, we now think that the Black Death was caused by Yersinia pestis or a variant thereof of this bacteria. And the Black Death was so problematic that even dealing with the death was something that a lot of people didn't want to do. Now, it did occur a few hundred years ago, and at this point, we might say that, well, they didn't have the modern antibiotics that we have. Penicillin had not been discovered. Vancomycin had not been discovered. And so, of course, it is possible that if such an outbreak were to occur these days, we would be able to tackle that because we have a panel of antibiotics. But before we let that kind of complacency take over, we just have to look at the trends in the introduction of antibiotics and the emergence of resistance to these antibiotics. And so penicillin was introduced in the 1940s, but it was introduced in the 1943, but it turns out that a penicillin-resistant bacteria was identified even before penicillin was introduced into the clinic. And so what we can see here is that it only took a matter of few years from the introduction of antibiotics to the identification of resistant bacteria. Methicillin is an important antibiotic because it is effective against staph, and staph infections actually occur on a large scale. But we can see here that after the introduction of methicillin in 1960, methicillin-resistant staph was identified only 2 years later. So the question is, does this resistant bacteria emerge from the introduction of the drug, or did it already exist in the environment and the drug just aided the expansion of this bacteria? The answer is that the bacteria actually already existed in the environment, and humanity, man, just aided the expansion of that bacteria. So at this point, we can almost make a generalized statement that whenever we develop a new antibiotic that kills bacteria, there will be resistance, and the resistance will be due to maybe a clone that already exists in the environment, or it could be due to the selective pressure and the mutation frequency in bacteria that would then lead to the emergence of a new resistance. And so we can visit this statement, and we will now say that with this data, the time has not come to close the book on infectious disease, and the time may never come for us to close the book on infectious disease. And so what I did before this talk is that I went to the Centers of Disease Control's website to really look at the problems of antibiotic resistance bacteria in the US. Not the world, but just the US. Okay. And so you can see here that globally, there was over 21 million Salmonella infections, right? I mean, that is mind-boggling. 21 million people were infected with Salmonella. Now, luckily, very few died. And so at this point, we could say that Salmonella might not be a problem. However, the fact that the bacteria can infect 21 million should give us reasons to be concerned. Because what if the Salmonella now becomes multi-drug resistant, right? It looks like Salmonella really can infect humans. Now, when it comes to the non-typhoidal Salmonella, we can now see here that not only does it also cause a lot of infections, but the cost to society is huge, $365 million. And then, you then have strep pneumonia. Again, 7,000 deaths in the US, which might not sound a lot, but then it costs close to 100 million. And then also, what is very interesting is that over 1 million people actually get infected with this bacteria. Clostridium difficile is a problematic bacteria. It infects 250,000, and it actually leads to 14,000. So now we are now talking about more than 10,000, and then it's becoming problematic. And it costs this much. The reason why Clostridium is problematic is that typically when people are infected with other bacterial strains and after they've taken antibiotics, and the antibiotic clears their natural bacteria, Clostridium can then easily take over and colonize. Okay? So this is also really problematic. And these are problems that are occurring now. We are not even talking about the future. Now, when it comes to gonorrhea, these numbers are really uncomfortable numbers. 246 for a sexually transmitted bacteria that can actually lead to reproductive issues later on in life. Okay? So these are all happening. These are not things that are in the past. And then you've got Shiga, etc. So I think that this gives you an idea that at this moment, we have problems with bacterial infections, but also we have problems with drug-resistant infections. And so the typical drug-resistant bacteria that we would come across these days would be maybe bacteria that will be resistant to one drug, And so the idea is that if you find a bacteria that is resistant to one drug, then you can use the next drug. There are over 30 antibiotics that have been approved for use. And so with 30 drugs, maybe I'm scaremongering everyone. Maybe we don't have a problem, okay? But we are just going to look at the facts, and then we will make a decision whether this is a problem or not. So if we look at this graph, what we see here is that the resistance to the traditional drugs, such as fluoroquinoline, seem to be increasing recently. So what I do find quite troubling is that for a very long time, these gonorrhea were susceptible to fluoroquinoline. And then around 2012, we could see a spike where resistance then went all the way up to 15%. Now, Salmonella, as I showed, actually infects a lot of people, right? And we can also see that gradually, Salmonella resistance to Cipro has gone up, that by 2011, up to 70% of Salmonella infections were caused by bacteria that were resistant to this Cipro, okay? But again, we have several antibiotics, so we don't have to worry about that. But if we think that the drug-resistant antibiotics that we have now, the bacteria that are resistant to the current antibiotics, is problematic, we've got to wait until the bacteria become multi-drug resistant, right? So what I mean by multi-drug resistant is that a possibility that there could be an infection caused by any particular strain of bacteria that will be resistant to all 20 to 30 antibiotics that are used in the clinic, right? This sounds ridiculous. How would that be possible? Surely there would be a bacteria that would be resistant to a few antibiotics, but it is almost unfathomable to think that there would be an infection caused by a single strain that will be resistant to all the drugs available to us that work on so many different pathways, right? Okay? Well, we will see whether that is possible, because if that is possible, then we all have to be worried, right? Then we are really heading towards doom, because what if these kind of strains then become, they evolve, and then they can easily affect all of us. So at this point, we should start to think about speeding up the development of new antibiotics, right? That if we are having issues with resistance, then there should be more antibiotics that are being developed. But if we now look at the number of antibacterial compounds that were approved by the FDA, we can see here that the number seems to be going down. Now, that doesn't make sense, because when I showed you the graph for the Cipro and the fluoroquinoline, we saw that the antibiotic resistance curve was going up. So why is it that bacteria that cause infection are becoming more resistant to our drugs, but somehow we are not developing as many? So I think I know why. The reason might be due to financial considerations. So it takes several millions, sometimes over 100 million, for a drug company to develop a new drug. You've got to do preclinicals and clinical trials, and then after that, you would hope that your drug gets approved. And if it gets approved, then you would hope that you'll be able to at least recover the amount of money that you've put into the development of that drug. But from what we've seen, it is very clear that bacteria would always have ways of developing resistance. And so it doesn't make financial sense to put so many millions of dollars into the development of something that will ultimately fail. And so we are seeing that, right? We are seeing that there is not a willingness to actually invest in the development of new antibiotics. So before we move on to talk about different strategies to solve this problem, I want us to really try to understand what is bacteria and what causes bacterial infections, because we tend to think of bacteria as some foreign entity that occasionally infect us, right? But it turns out that we are bacteria. So there was a study that was done a few years ago that showed that there is about 10 bacterial cells to 1 human cells, right? So that would mean that we are 90% bacteria and only 10% human. And then a recent study basically said that the first study overestimated the number of bacteria is actually 1 to 1. But I mean, that's still 50%. We are still 50% bacteria. Now, this really troubles me because I've always thought that, we as humans, we are a fixed entity, and our behaviors and everything that we do is fixed to the personality that was assigned to us at birth. But if we are up to 90% bacteria and these species are in flux, right? So they come into us and then they leave, it means that we are not as permanent as we think. We are actually beings in fluid. Maybe the Herman that existed a few years ago doesn't exist anymore, because the bacteria that colonized me a few years ago are completely different from the bacteria that are now living in me, right? So that is actually quite troubling to look at us that way. But also, what that means is that if we are bacteria, then how do we deal with this bacterial resistance and infection problem? Because it's almost like fighting self, right? It's almost like developing compounds to kill ourselves. The majority of these species actually coexist with us. They help us sometimes. They help us metabolize things. And then sometimes they are also doing really bad things, okay? So the Time and the Economist magazines and several other popular magazines have taken up to this. And so a few years ago, they basically had these covers that, microbes make man. We are more bacteria than human. And so therefore, when we are developing new compounds to deal with these bacteria, we have to have this in mind, that we could also be working against ourselves. Now, in addition to bacteria or us trying to kill us or them, now I'm being poetic, right? I don't even know whether we are bacteria or not. Bacteria could also be foes. So there are instances where it's been shown that bacteria causes Crohn's disease, right? Okay. Ulcerative colitis is an inflammatory disease, and that's all due to bacteria, okay? So they do a lot of things. And then, recently, it was also shown that bacteria could be linked to Alzheimer's disease, right? Because if bacteria causes inflammation, the immune system does its work very well by recognizing pathogens, and in recognizing pathogens, the immune system will then basically come up with robust response, but sometimes the immune system overworks itself and causes inflammation, and inflammation leads to a lot of damage, and neurodegenerative diseases could be linked to bacteria. So now we have, Crohn's disease, Alzheimer. Another study also linked Alzheimer to bacteria, where we see here that the beta amyloids pathology could also be linked to bacteria. And this was a very classic experiment where they actually had mice with bacteria and mice without bacteria in their guts, and they were able to link that to the pathology of Alzheimer via the beta amyloids. And then whenever we talk about cancer, we all think like, oh yeah, cancer is this horrible disease. It kills a lot of people, and we got to fund cancer research and put so much money into coming out with new cancer drugs. And it's now also emerging that bacteria can also contribute to cancer. So colon cancer, for example, has been linked to bacteria. So now, I'm getting confused here. We have the species that causes so much havoc to us, becoming drug resistant. It turns out that 50 to 90% of us are also bacteria. They link to cancer, Alzheimer, and everything. So is it time to just give up, right? And just live with bacteria and say, it is what it is. There's nothing that we can do. So recently, the World Health Organization published the first ever list of antibiotic-resistant priority pathogens. So now, the authorities are saying that we've got to basically pay attention to these, okay? So we have critical list. Gram-negative bacteria is becoming highly problematic. And so that is something that is really critical. But the high list is also including some of these gram-positives like staph. So methicillin resistant, remember, it started in the 1960s. And several decades later, we are still talking about this resistance. So what it means is that during these decades, we haven't solved that problem, right? The problem has existed. In 2017, we still have problem with methicillin resistance, and salmonella is also going to be problematic. So we, or the Sintim group here at Purdue, we have been interested in dealing with some of these. We don't have the resources to look at all the bacteria, but we've been interested in some of these, and especially we've been interested in Staph aureus, because I believe that Staph aureus is going to become really problematic in the future. Okay, so we've been interested in Staph aureus, and I'll show you some data in our lab. So why staph? Now, 80,000 infections in the U.S. are caused by drug-resistant staph. So this is not just staph infections. These are staphs that are resistant to different drugs. And out of that 80,000, 11,000 actually result in death. Okay, so that is very, very serious. Now, staph causes skin and wound infections and pneumonia, but sometimes you also get sepsis and death due to inflammation that leads to from the bloodstream infections. And so the first question that we want to ask is, can the antibiotic resistance be reversed? Because we know that we have several drugs that used to be effective against this pathogen. Can we come up with a new drug that will make the existing drug work again? Okay, so can we resurrect the older antibiotics? But so far, what we've neglected is the host response to bacteria, right? So at the beginning of the talk, I said that we are bacteria. We have different strains of bacteria coexisting with us. And during evolution, our immune system learned to deal with some of the resident bacteria, and so our immune system does not react vigorously to the resident bacteria. But when the pathogenic or the non-resident bacteria get in us, the immune system reacts, and of course they are doing their real job, right? They should react to the pathogen. But sometimes the reaction is too much. And that is what leads to inflammation, and then sometimes you can die from septic shock, right? So basically, yourself is killing yourself. It's not the actual bacteria that's killing you. It's actually how you react with it. And so in addition to dealing with this antibiotic resistance issue, there should also be strategies to find drugs that would actually calm down the immune system, right? But here is a caveat. The immune system is basically the soldiers that guard us against all kinds of infections. And so if we are to develop drugs to calm the immune system down, we got to be careful, because we might also start dying from some viral diseases that used not to kill us, right? You know, the flu epidemic, the flu could now start to kill us, and so many different diseases. And so there are issues here that we also need to start to think about. And then they always say that prevention is always better than cure. And so is there a way that we could even stop the bacteria such as MRSA from colonizing us, right? Now, unfortunately, I have news for you. For MRSA or for some staph, this is not going to be possible. A significant fraction of us are already carrying staph. And it is very difficult for us to stop staph from infecting us because staph co-evolved with us, right? So if we now look at this evolution tree, we can see here that the period that, we can see here that the period that the staph started diverging, right? So you had a parent, and then you started having divergence. That period coincides with when primates also started to diverge. So that's not coincidence, right? We have been co-evolving for a very long time. And so what that means is that it is very difficult to actually prevent having staph not colonizing us. So that anti-colonization strategy might be really problematic. So here are some of the uncomfortable facts about Staph aureus. Normal staph is present on the skin and the nose of about 20 to 30% of adults, right? So there are adults here. A significant number of people sitting in this room are carrying staph, okay? And then an estimated 1% of the US population are carrying drug-resistant MRSA without showing any symptom. So these people are going to be carriers, right? They might not be showing the symptom because maybe they have strong immune system, but remember that we carry staph on our skin, our noses, and so you've always got to be careful that a night out and meeting someone and a snog could lead to you getting methicillin-resistant staph, okay? So, you know, it's funny, but it's not funny, right? Okay, so now, let's now talk about solutions, right? We've been talking about the problems, but there has to be solutions, okay? So here are some solutions to the antibiotic-resistant problem. We have been developing drugs against various targets, right? I did say we have between 25 to 30 antibiotics in clinical use, and so these drugs attack the cell wall, the DNA, different proteins in the cell. And so we can continue to develop these drugs as we've been doing in the last decade. But we know that these drugs would meet resistance. And so the resistance mechanisms are known. The drug could come in and bind to a protein, but the protein could basically mutate. So the drug doesn't bind anymore, right? Let me go back here. The drug could bind to this protein, and maybe there is this residue in the protein's active site, but now the protein would bind to the drug, but the protein now has mutated to something else, right? And so the drug cannot bind anymore. Let me go back again. So initially, you see that this site had a positive charge, and so if the drug is negative, it can bind, right? And all it needs to do is to mutate. Now you've got a negative. It doesn't bind anymore, right? Very simple. So there are different ways the antibiotic could come in and could bind to the bacteria target, but the bacteria can basically cleave the antibiotic, right? And so many. Finally, I like this strategy. So if someone comes into your home at night, a very simple way to prevent that is to have video cameras, right? So that as they are approaching your house, the video cameras will basically sense them. And then they get into your house, and then, or maybe the video would prevent them, or it would scare them from getting into the house. But a very crude way is not to spend too much money on videos, right? It's just to wait until they get into the house, and then, you know, you hold them by the neck like this, and then you just flip them over the fence, right? Okay? So, and then maybe if you flip them hard enough, they won't do that again, right? They won't come in again. So that's basically what bacteria is doing here, right? The drug gets in, and the bacteria has got a protein. It's called an efflux pump, right? It grabs the drug and just throws it, right? So by and large, this is an ancient mechanism. The bacteria will not even have to know about what you are making. All it knows is that this is a foreign drug, right? It just holds it by the neck over the fence, okay? So they've ascribed this to Einstein, but my understanding is that the source is unknown. But they always say that insanity is doing the same thing over and over again, and then you then expect a different results, right? So we've been making antibiotics that target proteins, and resistance emerges, and so let's spend the next 50 decades doing the same thing, right? And maybe if we are lucky, something would happen. So why don't we come up with a whole new way of dealing with bacteria? Why don't we now accept that bacteria are part of us, the immune system is part of the problem, they could also be part of the solution. And so the new paradigm is basically looking at how bacteria come together to form biofilms. And when they form the biofilms, it's very difficult to kill them because bacteria in a biofilm are going to be about 10 times more resistant to antibiotics. And so the question is that can we come up with an antibiofilm drug to stop bacteria from forming biofilms, right? You've also got an immune system that is always dealing with the bacteria. Could we enhance the immune system, but could we also tame the immune system so that inflammation does not become so problematic, okay? So we got to think about biofilms, we got to think about the immune system, and we got to think about different ways. Now, bacteria also infect us by engaging with the receptors on our cell surface and then, putting in, I like this. This is like a needle, right? It engages with the host cells and then it puts a needle through the cell and then it pushes toxins through that. And so can we now find a small molecule to stop this needle from even assembling, right? Okay, so these are different strategies that would actually help us deal with bacteria. And then we now also know about bacteria signaling inside the bacteria cells. Can we come out with new molecules to actually stop the signaling? And so bacteria talk to each other via a process called quorum sensing. So you can see here that as bacteria population increases, the small molecules that they use to communicate with each other also increases, right? So bacteria, they do have a language. Now, if you work for some secret agency and you want to understand your enemy, I think a very smart way to go about it is to probably learn their language, maybe dress like them, get into their environment, and then you can get information, but also you can trick them, right? So language is critical. So if we understand bacterial language, then maybe we might be able to deal with bacteria. So a few years ago, we showed that this molecule, which is like a bacterial signaling molecule, right? It's synthetic, we made in the lab. The bacterial signaling molecule was this pattern, so we added this part to it. So we made a synthetic molecule. We could use this molecule to prevent bacteria from forming biofilm. So here is bacterial biofilm. We add our molecule, and we can actually reduce bacterial biofilms, right? So I did mention that biofilms make bacteria up to 10,000 more resistant to antibiotics. And so therefore, if we can stop biofilms, then maybe we might be able to deal with this resistance problem. Secondly, we are also interested in bacterial signaling, chemistries that go inside a bacterial cell. Here too, we think that chemists can make fundamental contribution and use molecules to disrupt these signaling molecules, right? We are almost like jamming how bacteria communicate. And so that would also lead to advances where we could start to disrupt biofilms or how bacteria actually talk to the host cell. Now, let's turn back to the immune system, because the immune system sometimes can become over anxious and do a lot of things. But it also turns out that bacterial signaling molecules can also affect the immune system. So this was a brilliant study that shows that, so LPS is basically molecules found on bacterial surface. So when they added LPS to immune system, what we can see here is that this is normal immune system, right? You can see that they are nice shaped, right? And then they added, this is a bacterial signaling molecule, and look at what is happening. The bacterial signaling molecule is basically making the immune system go like splash, right? And so these bacterial signaling molecules are also affecting our immune system, meaning that we need to come up with new ways of actually stopping that from happening. So the bigger picture here is that as bacteria are infecting us, and they are sometimes some of them get into the bloodstream, and our immune system gets alerted, and they start making all kinds of molecules to deal with bacteria. Sometimes the bacteria can also get into the cells. And so in my group, in collaboration with some of my collaborators at Johns Hopkins University, we've been interested in how bacteria such as TB that have also co-evolved with us for about 40,000 years and very successful. So TB can infect immune cells such as macrophages. We've been interested in how come TB is so successful. And it turns out that, we published this paper this year, it turns out that when TB gets into the human cells, as the human cell is trying to react to that TB, the TB also produces an enzyme that deactivates the human cell. So let me go back again to the statement I made about someone coming into your house and now you have a camera system, right? You decided that you were not going to hold that person and throw that person out because it is possible that it might be stronger than you. So now you have a camera system and your camera system will just detect them and then send an alarm to the security, right? And you might think that's a nice way to prevent anyone from invading your house. What if that person has a way to just turn off your camera system, right? And that's exactly what a TB is doing. The TB gets in, the host wants to detect the TB and then turn on cytokine production. And what the TB does is that the TB produces an enzyme that switches that off. And so the TB would be happy in the cell and the host would have no clue that the TB is in there. And so again, these are newer targets that we could actually start to look at. And so over the years, we have been looking at new ways to deal with this bacterial resistance problem by not looking at those cytotoxic mechanisms, but looking at bacteria signaling and biofilm formation and cyclic dinucleotide signaling and how bacteria talk to the immune system, because we believe that that is going to be the new generation antibiotics that would work. Okay, now let's get back to some of the scenarios that I think are already happening. And so we know that what is already happening is that we have vancomycin-resistant bacteria that are infecting a lot of people in the US and globally and leading to about, I would say, 1,000, right? So 1,000 might not be a lot, but about 5% death rate, okay? Now, I did mention that staph evolved with us, so it's very difficult to get rid of staph, right? Okay? So, if we now look at staph, vancomycin-resistant staph, only 13% cases in the US. But what if the number of vancomycin-resistant staph increases, right? Then that could be problematic. So that is already happening. I talked about the Black Death, right? The plague. We could say, well, this is an ancient disease. But if you go to the World Health Organization's website right now, in 2015, there were instances of cases caused by plague bacteria that led to 584 deaths. What if the plague bacteria became multi-drug resistant and then evolved to spread easily, right? So that could be problematic, okay? Now, at the beginning of the talk, I said, is it possible that we would have a scenario where there would be a species that would infect us, that would be resistant to all the drugs that we have, right? This is not a futuristic thing. It's already happened. In 2017, a woman died, and when they tested 26 different antibiotics against that bacteria, the bacteria was resistant to all 26. So it's already happened. The only thing now is that if it starts spreading, then we are in trouble, okay? So that is the doom. It's also been estimated that if we don't solve the antibiotic resistant problem in 2050, 10 million people per year will die from antimicrobial resistance. This is not just infections that are resistant to antibiotics, right? Now, if in 2017 we are already finding bacterial strains that are resistant to all the antibiotics that we have, then this is possible, okay? Now, there are two instances that have also been really bugging me a lot. So we now have technologies to engineer a lot of species, right? What if some crazy entity or regime is actually engineering a bacteria? I'm not talking about a bioweapon. I'm talking about someone engineering something like staph that gets introduced into the public, right? Not weaponized, okay? That is going to be really serious. Or what if the bacteria that caused the plague is already evolving in nature And all that needs to happen is for that to jump to humans, right? Are we going to be ready? Okay. Now, I will end this by saying that we can have global partnerships to accelerate the discovery of antibiotics, but we have to move beyond the traditional profit model because it's not going to be profitable, right? There is a problem that's going to happen. And then I also want to say that, immunotherapy, CRISPR-Cas, and so many advances in sciences now means that maybe when the time occurs, we might be ready, right? Might, okay? Now, the work that we do in our group would not be possible without the generous funding of the NSF, the NIH, and ABBA funding, and also Purdue University, as well as my former employer, University of Maryland College Park. And thank you again for coming to this talk today.